Hello everybody, my name is Jung Kim. I am studying at Professor Jung Kim's lab. Today I will tell you about MPBT, New Plant Breeding Technology. The title is MPBT, Molecular Breeding, Genome Editing of the Lateral Suppressor and Parthenocarpic Genes in Tomato. Introduction. Once upon a time, old days, people hunted and collecting fruits. But now, people are doing cultivation, farming. But in the future, we can expect that still we are going to do the farming, but many researchers will be the gene hunter. This is the new era of plant breeding. So conventional breeding, as we know, Mendelian rules, it was hybridization, and genetic modification, transgenesis between the different species, and cisgenesis between the same species. Genome editing is using CRISPR system now. So molecular breeding tool, CRISPR system, comparison of Cas9 and CPF1. We can call CPF1 Cas12a. So here the big difference. The longer trans-activating RNA and CRISPR RNA is in Cas9, and at the CPF1 you can see it's only the short CRISPR RNA. And POM also 3 nucleotide NGG POM site and 3 nucleotides away blunt end it makes. Differently, CPF1 TTTV usually we use as POM. So away from POM site, sticky end it makes. The origin of tomato. Wild tomato, very small, smaller than cherries. This started in South America. Now we are using MPBT. We can make ornamental tomato, experiment material tomato, edible tomato, and so on. New plant breeding technology. With this technical application, we can expect economic benefits, very practical. So horticultural management, this will be very easier than before through this genome editing at molecular level. What would be the economic benefits? First, reducing labor and cost. We can improve cultivation methods. Second, reducing nutrient losses. We can optimize nutrient management. Third, increasing market yield. This doesn't mean just make much yield at farming. This means for quality control. Approaching the target gene. When we look for the target genes, how can we focus on what trait kind of the things? Horticultural factors. We can see light quality and quantity, photoperiod, temperature, CO2 concentration, nutrition and water, pest and disease control. So when we think if we do tomato farming, or when we are looking for the tomato target gene, how can we find? We should focus on tomato architecture, trializing, removing old leaf, pedicel abscission, pollination, axillary shoot, pinching, hormone treatment, grafting, microbiome injection, Spring chemicals. If the tomato architecture is very good, this will be helpful for photosynthesis. Trializing. When we make trializing method much more simple, we can reduce the laboring cost. Also, those removing old leaf, pedicel abscission, and axillary shoot pinching, we can save much labor. In the case of pollination, without pollination, if we can get enough fruit, that could be very economical. How about grafting? To make disease resistance, people do grafting, but if we find the proper resistance gene, we don't need to do grafting, so we can save much money at farming. Also, for the microbiome injection, this is related to disease resistance, as same as spraying chemicals. So my experiment of MPBT, I'm interested in this LS gene, AGL6, and IA9 gene. So first, LS gene. You can see we call those the suckers. When we do this tomato farming, we must pick this axillary shoot. So among many genes, what I am interested in, these are lateral suppressor gene. This is the sucker. So there was a paper, automated pruning of greenhouse indeterminate tomato plants. To reduce this laboring cost, this team made the robots using phenotyping AI. So like this, they collected the samples and they made the program. So you can see when we do cooking, we discard these jelly seed parts. So I will tell you about these partner copy related genes. Tomato processing byproduct is big problem. When we can reduce these jelly and seed parts, only the fruit people usually eat, so we can save the money and energy and environmental friendly tomato we can make. So lateral suppressor gene is 1.3 kb, only just one single exon it has. What I'm interested in is at this part, the exon part, and also 3' enhancer part here. So this CDS region, when I make the mutant, it will make very strong, suppressing the axillary shoot initiation. And apart from the CDS 3' region, here, when I make mutant here, this will make very weak allele. So I'm using CRISPR CPF1, CDS region using 2 guide RNAs, and this 3' enhancer part, I use 4 guide RNAs. So expecting result is reduction of axillary meristem initiation. So the positive thing is, if the tomato plant doesn't make axillary shoot, we can expect that the much easier airflow, so this will reduce the pathogen attack. Very good for photosynthesis, and we can save nutrient. But the negative part is lateral suppressor gene mutant makes abnormality of floral organ and the reduction of fruit yield. You can see this is the wild type, this is the mutant. And floral organ also, big difference you can see here. This is my key paper. Tomato 3' promoter part, when they switch the place, it makes the difference of axillary shoot initiation. So yellow part means no axillary shoot, and greener part means every node has the axillary shoot. This is the younger stage, and it grows into this downward. So I got the idea from here. When I target this tomato 3' promoter region, I can make those kind of weaker layer. Sometimes it makes axillary shoot, sometimes it doesn't. So for agronomical traits, it's very good to get the enough fruit, and also some part of reducing the nutrient energy and labors. This is the 3' region. Just very short region. So to target the guide RNA part, I had some difficulties, but finally I found one from here and the other, and away from here some, 1 and 2. So I got 4 guide RNAs around this region B, 3' enhancer part. Next, I will tell you about parthenocarpy. As you can see here, parthenocarpy has no seeds. So when the plant suffer very severe environment, still the fruit we can see, but different from the proper condition, it makes no seeds. This means the fertility became weaker, and then it makes only ovary growth. So in real, consumers want this seedless fruit, but the seed company, they want the progeny, so they want this non-parthenocarpy fruit. So sometimes have seeds, sometimes doesn't have seed. When we make the balance, we can get the very good target. This is the key paper for the first parthenocopic gene, Agamus-like 6. This is 5.1 kb and it has many exon parts, so negative strand. If I can make the first exon knockout, I can make. So I looked for the proper locus for guide RNA. In this case, I used Cas9. So exon 1, 1 guide RNA, and exon 2, 1 guide RNA I applied. This AGL6 gene has very special expression. Only in the flower meristem, flower bud, and open flower. So, except those flower organ, every other part just very normal as wild type. You can see all looks same. Only the flower petal a little bit, you can see the curve. The viability of pollen also almost same. Fruit size and fruit weight also same, only you can see the difference. The seed here, no seed. Very interesting AGL6, so very useful to apply to lateral suppressor, which has the negative aspect of abnormality of fruit organ. So under the heat stress, this AGL6, you can see the much yield than wild type. So this makes fruit well under the severe environment. Non-emasculated, emasculated, or mutant has very smaller size seeds, and hetero it shows the wild type phenotype. Now the second IA9 parthenocopic gene. We know very well IAA, auxin. So this gene, auxin suppressor gene, size is 4.1 kb. So it starts from here, and also using Cas9. So at the first exon we got the guide RNA locus. So from here we can cut using Cas9 and it will be knockout mutant, wild type, and this is IA9 mutant. You can see the big difference at the phenotype. So the most different thing is the leaf shape. Those kind of leaf shape you can see here, and also the floral organ, just the ovary part is very huge. So here it's before the pollination, already ovary swollen like this. This means without the seed, only ovary becomes big. So no seed, because there's no fertilization. This IA9 mutant has shown those kind of 3-leaf cotyledon. And Carlix is wild type, looks very pretty, like star, but in the case of IA9 mutant, it's just a dumb shape, like potato leaf. So already we got the entire mutant through EMS. So after we did this IA9 mutant, we found that the one of knockout mutant was same as entire mutant. So we found one kind of mutant at IAA9 was same as EMS entire mutant. So here, this is IA9 mutant, and this is entire mutant. So when we look for the gene at Soul Genomics, you can see the IA9 mutant, the name entire. So lateral suppressor gene makes tomato architecture different. And parthenocopic genes, without the hormone treatment, we can get the result. So those technical application, we can get the economic benefits. Like much easier for trializing, so axillary shoot pinching, pedicel abscission. So LS mutant, using this CDS region, we can make strong allele, and the 3' enhancer part, we can get the weaker allele. And parthenocopy related genes, AJ6 and IA9. So my experimental goal is precise genome editing using CRISPR-Cas system to apply weak allele to confirm each gene function as to get the phenotype as normal as wild-type tomato and to maintain the yield as much as wild-type tomato. This is my hypothesis. This LS gene makes abnormality of floral organ. To avoid this, I applied AJ6 and IA9 because those two makes parthenocopic fruits. So in the case of IA9, there is just like a visual marker, we can see the abnormal leaf morphology. But this can cause the growth hindrance because it makes much shadowing at the growing stage. So my future plan, studying this MPBT, now I'm doing lateral suppressor and 3' enhancer part. I call this region B, and AJ6 IA9, but in the future I can find more genes. So the order is the first, designing construct and using agrobacterium transformation. Also the transformation method can be much more, we can make progress. So now using agrobacterium delivery, then I can make G0 plant phenotyping and genotyping, and then G1 generation through selfing. To doing segregation, I can get G8 to G10 and then I can make inbred line, and using molecular marker, CAPS marker, SCAR marker, SMP marker, I can do the gene stacking using CRISPR system, and then commercialization. So this is ongoing tissue culture. So to do the agrobacterium delivery, sowing and germination, cutting the cotyledon and pre-culture, agrobacterium transformation and washing, and shoot induction. After shoot inducted, move to another media for shoot differentiation. Among those shoots, when I see the meristem, I can move to root induction. After this, I can move to soil, and at the acclimatization stage, I can move those plants to the house or outside farm. Now my experiment is at the stage of root induction. As I told before, this is the same future plan. Thank you.